are there and many products are there. So from this chapter, maybe in certain cases the matching bits also given, from this chapter. So this chapter is very important, and we know about the microbes. Microbes can grow and at any place on the earth. See, especially either in the cool place, means especially either in the ice region, either at the high temperature, somewhat some organisms are grown at that -4, -5, -6 and 0 degrees centigrade, right? And some of the microbes can grow at 100 degrees centigrade. So the microbes have the capacity to grow at the different temperatures and different regions also, right? And these microbes, okay, here microbes means here not only the bacteria, some of the fungi can cause the diseases, and some of that microscopic organisms are there, microscopic animals are there, and microscopic organisms are there, and these also causes the diseases. See, if we compare the macroscopic organisms and the microscopic organisms upon the earth, right? So definitely the plants are there and the animals are there, right? Human beings are there. Okay, we are belongs to that macroscopic. Okay, we are one side, that is the macroscopic side, and there is a microscopic organisms also there. See, if we compare these two, the microscopic is the key role. The microscopic organisms, okay, is the main key role, because which performs the different types of functions. So here there is a microbes are there, the microbes, there is a bacteria, some of the fungi are there, and some of the other types of microscopic animal creatures also there, right? So see, generally we can't see the microbes directly. That is nothing but by the naked vision. When we have the microscope, that time we can see. And one more chance is there, when these microbes which are cultivated within the laboratory, especially within the nutrient media, right? When we have the Petri plate and if we pour the nutrients after the pouring process, if we streak some of the microbes which forms the colonies at the surface of the nutrient media, right? So especially there is an individual colonies are formed, then the individual colonies are nothing but microbes. So we can see that colony structures, right? So see, sir, why we are forming that colonies, or we are cultivating the bacteria? See, all the bacteria are not only all the bacteria are not harmful. Some of the useful bacteria also there. This chapter is going to deal with that useful bacteria, especially within the human life, or beneficial bacteria or beneficial microbes, especially beneficial microbes to the human beings. We are going to discuss in this chapter. See, we know that one of the lactobacillus bacteria is there, right? The bacteria is very useful to the human beings, because we are eating curd. The curd is produced by the lab. Lab is nothing but lactic acid bacteria. We know in the first year also we discussed about the curd formation, right? So how the curd is formed. Okay, one of the milk is there. The milk is subjecting for that pasteurization process. First suppose this is one of the milk. The milk is subjecting for the pasteurization process, right? Once the pasteurization is completed. So what is pasteurization? We know when the milk temperature is raised, okay, too high, means one of the container is there. We pour the milk within that container or the bowl. We are raising the milk temperature. Up to 70, 80 or 90 degrees centigrade, and immediately we are cooling down the milk temperature, because when we raise the temperature, that time some of the microorganisms are killed. Maybe certain cases some heat resistant bacteria are also there. So these are killed when we are immediately cool down the milk temperature. So here, by the pasteurization process, okay, the milk sterilization, that is nothing but killing of microorganisms. That is nothing but pasteurization, right? So after the pasteurization process, we are adding that some of the curd, very minimum amount of curd we are adding to the milk. That is nothing but inoculum, the addition of curd, okay, little amount of curd, okay, that is nothing but inoculum. That is also called starter culture. So the presence of lactobacillus bacteria, how the milk turns into the curd. We know this concept. We already discussed whatever that bacteria are there. That bacteria, what are the bacteria means here? One of that lab bacteria, right? Lactic acid bacteria. That bacteria produces some of the acidic substances and alters the pH of the milk. Then whatever the casein proteins are there, that casein proteins are partially digested. So presence of bacteria, acidic substances are produced, and presence of bacteria, casein protein is partially digested. Then there is a curd formation which takes place. So here there is a casein proteins are there. The casein proteins are partially digested. The partially digested casein proteins which are bound to the calcium ions. Then there is a semi-solid state curd formation which takes place. Cold formation takes place, right? We know this is one of the lactobacillus bacteria. That lactobacillus bacteria, that is also called lactic acid bacteria. Right? This is also called lab. Remember, these are the other names of that lab. We have to remember, right? And which produces some of that acidic substances. Presence of these acidic substances and presence of these microbes. That casein protein destruction takes place. Casein protein destruction takes place. And remember, we know that curd formation is done by that lab. Not only that one, some of the other beneficial effects also there to the human beings. Remember, we know that increase the nutritional quality of the curd. So how the nutritional quality increased by the lab? See, vitamin B12 synthesis. So the vitamin B12 synthesis, especially done by the lab within the curd, right? So within the curd, vitamin B12 is there. Then we are eating the curd, then we are getting that vitamin B12. Okay? And not only this one, there is an Escherichia coli. E. coli is there. Okay? E. coli, that one is also present always inside the human body, especially our intestine. That one is also produces that vitamin B12, right? See, this is the only vitamin not synthesized in the human body, and this is also not available in other sources also, especially synthesized by the bacteria. That is nothing but one of the lab bacteria, and the other one is the E. coli. Okay, the main one is the E. coli, and this one is also synthesizes that vitamin B12, right? And see, the lab bacteria are also supporting to enhance the human immune system, enhance the human immunity, especially. Human immunity means, is there any other disease causing agents are entered into the human body that time? Okay, that organisms are checked, that disease causing bacteria are checked by the lab. Once these are entered and activates our immune system and supporting to kill that disease causing bacteria. Simple. If we eat the daily bacteria part, then we are getting the bacteria part means if we eat daily the curd, okay, that is nothing but bacteria cells, then we have certain immunity because daily we are eating bacteria, okay, that bacteria cells products especially enter the bloodstream, activates our immune system, right? So that is about the curd formation and the beneficial effects. And coming to the next one is that fermented products. Remember, here we are discussing about the household products, microbes in household products. We are discussing about some of the microbes which produces the different types of products, especially household products. Okay, we are discussing now the fermented products. I thought everyone remember, idli and dosa. Okay, see, dough is there. We know the dough preparation process. There is a black gram or the green gram, some of the pulses are there, and in certain cases cereals also there, right? And the pulses, black gram and the green gram, we are subjecting for the soaking process after that grinding process. we get the dough. The dough is subjecting for the for the fermentation process. The fermentation is done by that bacteria. Then results of bacteria. See, tomorrow if I want to eat the idli or the dosa today evening, we need to prepare that dough, because from today night to tomorrow morning, fermentation will be done by some of the bacteria. We are not directly adding what we are adding that water, what we are adding that other substances. That substances also present these microbes. Means microbes are presented everywhere, either in the water, either in the soil, and different regions. Okay, within the air also the microbes are present. Right? So that's the reason why. Okay, the dough is fermented by the bacteria. The dough is puffed up. Okay, the dough is puffed up with the carbon dioxide. So what is the use? What is the use of fermentation of dough? See, the concept is very simple. If we directly make idli and dosa without any fermentation, without any dough fermentation, we didn't get that proper idli and dosa. The dosa will be tear out into many number of pieces, and we are getting that idli, but maybe not made in the proper shape. Okay. Because of that, when the fermentation, if the fermentation is not takes place, we didn't get proper taste, and we didn't get proper idli and dosa, even the structure also, right? When the fermentation is done, that time, whatever that large complex structures are there, the large complex structures forms the converted into, after the grinding process, the large complex structures are converted into that small products. Then carbon dioxide production takes place. We know the carbon dioxide is the very important one. Once the fermentation is done, once the carbon dioxide is there, then we get the smooth idli and the smooth dosa, right? So that is about that the fermentation benefit for the making idli dosa. Not only this, we are eating bhaji also. Within bhaji, we are adding that chemical agents, right? We are adding one of that salt, right? That salt have the capacity to release the carbon dioxide. That time, okay, the bhajis are swelled. The bhajis, okay, are swelled. Presence of carbon dioxide. Right? So after this is about that products, and we are eating. The taste also enhanced here. And this is about the very general concept, and most of the students like this concept also do. And presence of the yeast, especially within the bakeries, bread baking process, and the cakes are prepared. Birthday cakes we know, and pastries also we know. So here the bread baking and the cakes and the pastries, all these are prepared by the fermentation process. Here the fermentation done by the yeast, right? And one of the oldest food in India, right, that is cheese. We know about the cheese. When the buffaloes or the cows produces that young babies or young cows, especially young cows, right, that time, at first time, okay, the cows and buffaloes produces that milk, right? The milk contains the different types of enzymes are there, right? The renin enzyme, that one is especially supporting for the production of the cheese. So we prepare the dough. Dough is nothing but here, not that cereal substances. Here dough is nothing but we are making one of the normal daily milk is there. The daily milk is substituting for that the first milk we collected. The first milk means here one cow is there. Cow produces the baby. Okay, first time the cow gives the milk. That milk is we are collecting. That milk is we are collecting. After that collection process, that milk we are adding to the normal milk, and we are adding the sugar. We are adding some other flavoring substances also, right? And we subjecting for the boiling, right? That is we produce the normal cheese within house, and within the different types of companies are there, especially some of the industries also there. Okay, in that case, Propionibacterium shermani, right? That is the one of that organism we are using to produce the cheese. That cheese is called as Swiss cheese. Remember that cheese especially produces the large holes. Okay, that cheese produces large holes. Sir, why here large holes are produced? Remember, when we are preparing that dough, that time we are adding that we are adding these microbes, right? See, these microbes are involved in the fermentation. During fermentation process, carbon dioxide is produced. That carbon dioxide, during the boiling process, produces the large holes. So, how these holes are produced? Presence of high amount of the carbon dioxide. Okay, the large holes are produced. Okay? That is about the dough preparation. That is about the Swiss cheese production. Right? And coming to the next concept is the ripening of the cheese. See, after the production of the cheese, the surface is treated. The cheese surface is treated with that different types of microbes or different types of ripening agents. The flavor of the cheese is altered. For suppose one of the cheese I already produced, but I want additional flavor. For that flavoring process. I added one of the fungi on the surface of the cheese. Then, okay, after that, addition process, sometimes I subjecting for that incubation. After that incubation process, whatever that surface and whatever that surface is there, the surface produces one kind of smell because of the addition of the flavoring agents. By the addition of the flavoring agents or addition of the ripening agents. The ripening agents are nothing but here fungi. See, here the dough, subjecting for the ripening, dough produces the cheese. For suppose this is the dough. Dough produces the cheese. This is the cheese. After that, we are subjecting for the ripening of the cheese. Here I added one of the fungi. Especially the penicillium species we are adding, and finally it produces that Roquefort cheese. Right? So that is about one of the Indian oldest food material produced by the microbes. And see, this is one of the household fermented drinks. I thought most of the people are now about this one. Expecting toddy. The product name is called as toddy. We know about that palm trees. Whatever that palm trees inflorescences are there, we are collecting sap. In Telugu, this is also called kallu. See, we know about the Borassus flavellifera. The plant name is Borassus flavellifera, right? That plant, especially the male plant, produces the inflorescence. That inflorescence are collected. The sap is collected. The sap is subjecting for the fermentation process. Right? Okay. After that fermentation process, that sap is nothing but toddy. The sap is nothing but the toddy. Right? So this is one of the traditional drink, especially in the South India. This is one of the traditional drink in South India. Right? That is about the toddy. I thought most of the people are well known and expecting about the toddy. And whatever that, some of the bacteria also we are using. For the fermentation of the fishes. Okay, we know some of the dried fishes. Okay, we are cooking. Some of the people are cooking, right? So how the dried fish is prepared? See, high salt concentration is required. At the same time, some of the bacteria also required. Presence of these bacteria, the fermentation of fish will be done, right? Then that one is having the other nutritional values also. Right? That is about the household products. And next one we will discuss about the industrial products produced by the microbes.